This conference will now be recorded.
Hello students, now we are learning about another important chapter of biology. This is biomolecules. This chapter is about biomolecules, and here according to NEET syllabus you have to learn about the chemical constituents of living cell, and in that biomolecule structure, their function of protein, carbohydrate, lipids, nucleic acid and all. Then about the enzymes we have to learn, and the properties of enzyme and the enzyme action, classification, nomenclature of enzyme. About all these we are learning. So this is an important chapter. Like from here you won't get like all direct questions only you'll get from this chapter. So let's see what and all we need to learn in this. So we know that about 99% of the substance of all living things, they are made up of 4 elements. So which are those 4 elements? Hydrogen, oxygen, carbon and nitrogen. Hydrogen, oxygen, carbon and nitrogen. These are the 4 elements, and all these elements, they are combining in different proportions, right? And in different ways. And that's how the biomolecules are forming. So their combination, the combination or the proportion of combination, that is different, and that forms the biomolecules. Clear? So biomolecules are the organic molecules which is present in all the living organism. This is present in living organism. Biomolecules are present in the living organism. So different types of biomolecules, then compounds, ions, all these which is present in a cell, that is called as cellular pool. Okay, so the collection or the sum total, we can say sum total of these compounds, the ions and the biomolecules, all these which are present in a cell, that is called as cellular pool. See different components of the cellular pool. We can say biomolecules. These biomolecules, we can tell these biomolecules in different types, like large molecules are there, small molecules are there. Large molecules and small molecules. Okay, this large molecule or large biomolecules, they are called as biomacromolecules. Biomacromolecules. And the small molecules, they are called as biomicromolecules. Okay, and this biomacromolecules or the large molecules, that includes protein. Examples, we can say protein, then nucleic acid, polysaccharide, lipid, polysaccharide or carbohydrate, we can say, and small molecules or biomicromolecules. For that, we can say primary metabolites and secondary metabolites. Primary metabolites and secondary metabolites. Okay, this primary metabolites, we can say they are directly involved in the growth and reproduction. That means they are the small peptides, enzymes, and all. They are called as the primary metabolites. And the secondary metabolites, they are indirectly involved in the growth process, like antibiotics, pigments, alkaloids. All these are coming as secondary metabolites. Clear? Next, we need to learn about all these biomacromolecules in detail. So here first we are learning about carbohydrate. See here only we are covering previous year 10 years question paper. From that which and all questions came from, only based on that we are explaining now. So only from those questions, previous year question papers, only those points we are covering now, so that it will be easy for you to learn the main points and the special points which is from previous year question paper. Instead of answering that, if this classes you are covering means 10 years question papers you are covering. Okay, so here we are learning about the carbohydrate or saccharides. So these are among the most widely distributed compounds both in plants and in animal kingdom. In plant kingdom and animal kingdom, these are most widely distributed. Which one? Carbohydrate. Clear? And these carbohydrates are defined as polyhydroxyaldolases or ketoses and their condensation products. Polyhydroxyaldolases. Polyhydroxyaldolases or ketoses and their products. Okay, and their condensation products. So this carbohydrate in their organic substances, they have carbon, hydrogen, oxygen, and here the oxygen and hydrogen, they occur in a particular ratio. That means 1:2 ratio. Carbon is there, hydrogen is there, oxygen is there. Here oxygen and hydrogen, that is occurring in the ratio of 1:2. In which one? In carbohydrate. Okay, so the general formula of carbohydrate, that is CH2ON. Okay, and on the basis of the products of hydrolysis, these carbohydrates are divided into three major groups, that is monosaccharide, oligosaccharide, and polysaccharide. Carbohydrate that is divided into three groups. Which are the three groups? Monosaccharide, second one is oligosaccharide, third one is polysaccharide. Okay, so let's see what is monosaccharide. Monosaccharide, these are simple group of carbohydrates. This is simple type of carbohydrate and this is containing aldehyde or ketone group. Which group? Aldehyde or ketone group. Okay, so these aldehyde or ketone group, it is containing and they are sweet tasting, they are colorless, they are soluble in water, but these are insoluble in alcohol and ether. Insoluble in alcohol and ether. Which one? Monosaccharides. And these monosaccharides can be further divided into trioses, tetroses, pentoses, hexoses, heptoses. So this is based on the number of carbon atoms. So monosaccharide, this is divided into, this division is based on number of carbon atoms. Okay, so based on number of carbon atoms, it is divided into trioses, second one tetroses, third one pentoses, Fourth one, hexoses. Then fifth one, heptoses. Clear? So here trioses means 3 carbon atoms, right? So for that, examples we can say dihydroxyacetone and glyceraldehyde. All these are the examples for trioses. What about tetroses? Tetroses means 4 carbon atoms will be there. So erythrose and triose. Next comes the pentoses. 5 carbon atoms will be there. So example is ribose, which is found in RNA. The sugar which is found in RNA. Next comes hexoses, which is having 6 carbon atoms. So example is glucose. Glucose is there, galactose is there, then mannose, fructose, all these type of sugars. So glucose, which is found in blood sugar, then galactose, which is component of milk sugar, mannose, which is common in plants, then fructose, that will be the sweetest of all sugar, which is found in fruits. So fructose is the sweetest sugar and that is present in fruits. Okay. So hexose means 6, it will be having 6 carbon atoms. Examples glucose, galactose, fructose, mannose, all these type of sugars, that is called as hexoses. Okay, next comes heptoses. Heptoses, it is having 7 carbon atoms. So example is pseudoheptulose. Pseudoheptulose. And this pseudoheptulose, that is an intermediate in Calvin cycle. So this is an intermediate in Calvin cycles. So that's about the monosaccharides. See here, these many points I mentioned, all these are previous year questions. Okay, that's why especially I mentioned everything. So all these are previous year questions in different years. All these questions came. So next is about oligosaccharide. Oligosaccharide means, oligo means few, saccharon means sugar. So all these are the groups of compounds which are formed by the condensation of 2 to 9 monosaccharides. Here oligosaccharide means it is by the condensation, it is formed by the condensation of 2 to 9 monosaccharides. Clear? And all these monosaccharide units are joined with the help of some specialized glycosidic bond or glycosidic linkage. So these 2 to 9 monosaccharide units are joined by glycosidic bond or glycosidic linkage. An example for oligosaccharide, that is lactose or milk sugar. Okay, so this lactose or milk sugar, this is present in the milk of mammals and this is made up of 1 glucose and 1 galactose unit. 1 glucose, 1 galactose unit. Which one? Lactose. So that is present in the milk of mammals and this lactose is made up of 1 glucose molecule and 1 galactose, 1 glucose unit and 1 galactose unit. So this lactose, that is a reducing sugar. The same portion, about all these you will be learning in chemistry also, 11th standard chemistry. So you can combine both these chapters and learn. Okay, so lactose, or it is called as milk sugar. Next comes maltose or malt sugar. So maltose or malt sugar, this is named like this because of the occurrence in malt grain of barley. So this is present in the malt grain of barley. That's why the name is given as maltose or malt sugar. Okay, so this is present in the malted grain of barley, and it is mostly found in the germinating seeds and tissues, where it is present in the germinating seeds or tissues. So there the starch is broken down in that. Okay, in such cases, maltose or malt sugar will be present. And next comes, which is the next one? Next one is sucrose or table sugar. Sucrose or table sugar. So this is called as, or sucrose is called as cane sugar or invert sugar. Cane sugar or invert sugar. Okay, so in this, in cane sugar or invert sugar, fructose occurs in pentagon form. Fructose in pentagon form. Hexose or the glucose, that is in hexagon form. And this is, or the sucrose is a known reducing sugar. Okay, so these are the points from the previous year question paper. Next comes the raffinose. Fourth type is raffinose. This is a trisaccharide. Trisaccharide, and that is having glucose, galactose, and fructose. It's having glucose, galactose, then fructose also. Clear? Next comes, or the fifth one is cellobiose. So the cellobiose, that is a disaccharide. Okay, it's a disaccharide, and this is having 2D glucose subunits. Which is having 2D glucose subunits, and it is the structural component of cell. Which one? Cellobiose. Structural component of cell. Clear? So that these are the points which we need to learn about oligosaccharide. Next, we are moving to polysaccharide. Here, polysaccharide, poly means many. So many sugar units. That is, it's a polymer. That means that is containing minimum of 10 monosaccharide units. Minimum 10 monosaccharide units will be there. Okay. So minimum 10 monosaccharide units will be there. So polysaccharides are of different types, like two types polysaccharides are there, homopolysaccharide and heteropolysaccharide. Homopolysaccharide, heteropolysaccharide. Homopolysaccharide means, or we can call them as homoglycans, and they have only one type of monosaccharide units. Homo means same type. One type of monosaccharide units, and some important monosaccharides, they are glucans, galactans, mannans, glucans, galactans, mannans. So glucans, they contain only one glucose unit. For glucans, we can say example starch, glycogen, cellulose, chitin, all these are glucans. Then galactans, we know that they have galactose units. Only they have galactose units. Okay. And example we can say agar, pectin, galactan from snails. All these are the galactans. Next comes the mannans. In mannans, we can say these contain only mannose unit. Mannans only contain mannose unit. Example, yeast mannans. Okay. So that's about homopolysaccharide. Next comes heteropolysaccharide. This heteropolysaccharide or heteroglycan, that is having at least two types of monosaccharide. Minimum two types of monosaccharide units. So example we can say chitin, pectin, peptidoglycan, mucopolysaccharide, all these. So major polysaccharides, which are the major polysaccharides which we need to learn? That is starch, glycogen, cellulose, chitin, all these are the major heteropolysaccharides. So that's about the carbohydrate. Next, we need to learn about proteins, another type of biomolecule, that is proteins. So the word protein, that was first coined by Berzelius. Okay, and Mulder. They coined this term, and this is involved in various functions like structural function is there, storage function is there, transporting and signaling will be there, then movement is there. So all these functions are there for protein. So these are natural heteropolymer, and the substances like amino acids. Okay. In biosphere, which is the most abundant protein? The most abundant protein in biosphere. Most abundant in biosphere, that is rubisco. Rubisco is the most abundant protein in biosphere. So first, the basic unit of protein, that is amino acid. So amino acid means the compound which is having both amino and carboxylic group. Amino group will be there plus carboxylic group. Okay, that's why it is called as amino acid. And this amino acid, they will link together through peptide bonds, and that's how they will form the peptide or protein. Okay, then the amino acid, that is classified on the basis of synthesis in the living organism. So amino acids are classified into three categories based on or on the basis of synthesis in living organism. That is essential amino acids, Second type is non-essential amino acids. And third type is semi-essential amino acids. Okay, these are the three types or three categories of amino acids which are synthesized in the living organism. So in this essential amino acids, we know that cannot be synthesized by any organism in the body. And these are to be obtained from the dietary sources. Essential amino acids from diet, or these are from dietary sources we need to get. So they are essential amino acids. There are 8 essential amino acids in the case of animals. And in the case of humans, there are 7 essential amino acids in the case of humans. Okay, and next comes the non-essential amino acids. This non-essential amino acids can be synthesized by an organism. So this is synthesized, non-essential amino acid is being synthesized in an organism, and this is not required as the dietary component. And these non-essential amino acids, they are 10 in numbers. Next comes the semi-essential amino acids. This semi-essential amino acid, which is required essentially by an organism during a particular phase of the body growth and lactation period. That means in pregnant mothers and all, this is required, semi-essential amino acid. And these semi-essential amino acid, they are two in numbers. Semi-essential amino acids are, there are 2 semi-essential amino acids. And on the basis of chemical nature, so based on the chemical nature, amino acids are divided into neutral amino acids. Amino acids, there are of 3 types: neutral amino acids, acidic amino acid, and basic amino acid. Right? So the neutral amino acids, they have 1 amino group and 1 carboxyl group with non-cyclic hydrocarbon chain. So for neutral amino acid, you have an example, glycine, which is a very simple amino acid, glycine. Then alanine, valine, leucine, isoleucine, all these are the neutral amino acids. Next comes the acidic amino acid. This acidic amino acid, they have an additional carboxylic group. Example, we can say aspartic acid, glutamic acid, glutamine, asparagine, all these are the example for acidic amino acid. Next comes the basic amino acid. So they have additional amino group. Which one? Basic amino acids, they have additional amino group without forming amides. Okay. So arginine and lysine. Both are the examples for basic amino acids. Then another classification, this first, now whatever we have discussed, that is based on the chemical nature, this division. And on the basis of side chain, amino acids are of different types. So on the basis of side chain, amino acids are first one sulfur containing amino acid, then alcoholic, aromatic, heterocyclic. Sulfur containing. Next is alcoholic, aromatic, heterocyclic. Okay, so all these are on the basis of side chain. Next, the classification of protein. Classification of proteins. Here, on the basis of composition, proteins are classified into two classes. First one is based on composition. It's classified into fibrous protein and globular protein. Okay, fibrous protein means their structural material. They are the structural material of animal tissue. Okay, so when the polypeptide chain run parallel and they are held together by hydrogen and disulfide bond, then fiber-like structure will form. Okay, so example we can see keratin, fibrin, collagen, myosin. Keratin is a fibrous protein, then fibrin, collagen, myosin. These are fibrous protein. Next comes the globular protein. So globular protein, they perform the regulatory function in the body. So what is the function? Regulatory function in body. And in globular protein, the peptide chains are coiled about themselves, and that is resulting in the spherical molecule. Which one? Globular protein. So for example, globular protein, we can say enzymes, hormones like insulin and hemoglobin. All these are globular proteins. Next, another classification that is on the basis of components of molecule, which is present in the protein. So based on that, based on the components of protein, protein that is classified into three classes: simple protein, then conjugated protein, third one is derived protein. Okay, simple protein means this is made up of amino acids, only with amino acids. Okay, so example we can say collagen of connective tissue, albumin, albuminamide, albuminate, then globule. All these are the simple proteins. Next, conjugated protein. Conjugated protein is made up of protein molecule which is joined to a non-protein part. Protein molecule plus non-protein part. Okay, this non-protein part is otherwise called as the prosthetic group. Example, hemoglobin, casein of milk, all these are a conjugate example for conjugated protein. Okay, next one is derived protein. Derived proteins are formed by partial breakdown of natural protein. Partial breakdown of natural protein. So it is getting derived from something else. That's why it is called as derived protein. So for example, we can say peptones, insulin, fibrin, all these are derived proteins. Insulin, fibrin, then peptones, all these are the examples for derived protein. Okay, so we have learned about the classification of protein. Next is about the structural level of protein. So structural level, there are 4 structural level for protein. Primary structure is there, primary structure, then secondary structure, tertiary structure, quaternary structure. These are the different structures or structural levels in protein. Okay, so these are the structural levels of protein. So quaternary structure of protein, that is, for example, we can say hemoglobin. Okay, that's the quaternary structure. So here we have covered carbohydrate, protein, and next which one will come? Next, we have to learn about lipids. This term lipid, that is given by Bloor. This lipids, chemically they are very diverse group of compounds, and these are characterized by their relative insolubility in water. Okay, but they will be soluble in organic solvents. So these lipids can be defined as the esters of fatty acid and alcohol. Esters of fatty acids and alcohol. Okay. So in animal kingdom, both in the animal kingdom and in the plant kingdom, this lipid is having wide distribution. So both in animals and plant kingdom. Okay. So that's about a lipid. Next, fatty acids. Here, fatty acid means this is water insoluble long chain hydrocarbons. Water insoluble long chain hydrocarbons. Okay. So the long chain in the sense 4 to 36 carbon length will be there. Okay. So that is having one carboxyl group. or COOH group. So this is a simple like carbon and carboxyl group. This is a simple constituents of lipids. Okay, so fatty acid chains that may be of different types like saturated fatty acid and unsaturated fatty acid. Okay, saturated fatty acid, unsaturated fatty acid. Next, this lipids. Lipids are classified into simple lipids and compound lipids and derived lipids. Simple lipids, compound lipids, derived lipids. So simple lipids, they are the true lipids. Here, examples we can say triglycerides, waxes, all these are simple lipids. Then fat, wax, suberin, cutin, all these form the fatty acid and this is from fatty acid and alcohol. So here triglycerides and waxes we can say so these are simple lipids, right? Next comes the compound lipids. Compound lipids this compound lipids they are otherwise called as conjugated lipids. Conjugated lipid uh, they have an additional group in addition to the alcohol and fatty acid. Additional group will be there. So for example, glycolipid will be there, phospholipid, lipoprotein, all these are the examples. So glycolipid, phospholipid, then lipoprotein. So lipoprotein, on the basis of their compactness, lipoproteins are divided into LDL and HDL. LDL means low density lipoprotein. Then HDL, that is high density lipoprotein. LDL, for example, low density lipoprotein that is otherwise called as bad cholesterol. LDL is called as bad cholesterol. HDL, that is high density lipoprotein that removes bad cholesterol. High density lipoprotein that removes the bad cholesterol. Okay, so now we have learned about simple lipids, compound lipids, and next comes about the derived lipids. Then comes the derived lipids. Derived lipids means these are the derivatives of lipids or steroids, or it's a chemical. So the derivative of lipid or its chemicals, that is called as derived lipids. Steroids, palmitic acid, all these are derived lipids. So here in this chapter now we have covered about biomolecules, in that carbohydrate, protein, then lipid. These are the biomolecules, and next we need to learn about the nucleic acid, DNA and RNA. So in that, the structure of DNA and RNA we need to learn. Then about enzymes also we need to learn. So both we will be learning in the next class, because that is a DNA and RNA, which are the nucleic acids. Here when we are saying about DNA and RNA, we need to learn about the structure of DNA and the nitrogenous bases which is present in DNA. So the Watson and Crick model of DNA. So that's a separate topic, like a continuous topic. And about RNA also we need to learn. Then enzyme. While learning about enzyme, we need to learn about simple enzyme and conjugated enzyme, the mechanism of enzyme action, chemical nature of enzyme action, classification of enzyme, then enzyme inhibition. So about all these things we need to learn. So about enzymes and nucleic acids we'll be learning in the next class. Thank you.